When we see ourselves in the mirror, we only see it as a human body. But what happens if we zoom in? Like, really zooming in. Hello, welcome back to Bubbly Bio. Today's topic will be IB Topic 1.1 Introduction to Cells. As many of you might already know, this channel is created for students struggling with biology, so just relax and feel comfortable while we go through the understandings, applications, and skills.
Okie dokie, let's start.
Understanding 1. According to the cell theory, living organisms are composed of cells. The cell theory states that cells can only arise from pre-existing cells, and all living life are composed of cells, which is the smallest unit of life. Although this cell theory applies to many life, we still can question it. Application 1. Questioning the cell theory using atypical examples, including striated muscle, giant algae, and aseptate fungal hyphae. Striated muscles are fibers formed by muscle cells fused together, which makes it a multinucleated fiber. This challenges the cell theory as the muscle cells are not functioning as independent units. Unicellular giant algae can be very large in size, which challenges the idea that larger organisms are made of microscopic cells. Aseptate fungal hyphae has no septa and has continuous cytoplasm, which challenges the idea of cells being the smallest unit of life. Understanding 2. Organisms consisting of only one cell carry out all functions of life in that cell. Organisms consisting of only one cell are called unicellular, and they carry out all 7 functions of life. First, nutrition, done by obtaining food and provides energy for growth. Second, metabolism. This is the chemical reaction inside the cell. An example is cell respiration to release energy. Third, growth. This is the increase in size. Fourth, response. This is the ability to react and to change in an environment. Fifth, excretion, done by removing waste products of metabolism. Sixth, homeostasis, done by maintaining a stable internal environment. Seventh, reproduction, done by producing offspring either sexually or asexually. Application 2. Investigation of functions of life in paramecium and one named photosynthetic unicellular organism. Since unicellular organisms live by one cell, all functions of life take place by one cell. In paramecium, a hair called cilia allows it to move, which is response. Paramecia engulfs food via cytostom, which is nutrition, and then the food particles are enclosed within a small vacuole where the enzymes digest, which is metabolism. As for excretion, solid wastes are excreted by anal pore, but liquid is pumped out by contractile vacuoles. Oxygen enters and carbon dioxide exits the cell by diffusion, which is homeostasis. Paramecia divide asexually, considered as a reproduction. Another example for photosynthetic unicellular organism is synedesmus. Synedesmus exchanges gas and materials by cell diffusion, which is nutrition and excretion. It also contains chlorophyll, producing organic molecules by photosynthesis, considered as metabolism. Parent cell divides and reproduces asexually, which is its reproduction, and lastly, synedesmus can exist by themselves or form colonies for protection, which is response. Understanding 3. Surface area to volume ratio is important in the limitation of cell size. Cells produce chemical energy via metabolism and require efficient exchange of materials, which means if the volume of the cell increases and requires increased exchange of materials, since the surface area does not increase as much as the volume, the cell will fail by metabolic rate exceeding the rates of exchange. Thus, growing cells often divide and remain small to survive. Additionally, cells and tissues targeted for exchange of materials and gases often have ruffled structures to optimize material transfer. As an example, villi in intestinal tissues and microvilli in alveolar cells. Understanding 4. Multicellular organisms have properties that emerge from the interaction of their cellular components. Unlike unicellular organisms, multicellular organisms are capable of synergistic effects created by collective actions of individual cells. Cells form tissues, multiple tissues come together and form organs, then the organs interact, creating an organ system which carries out functions of life. Understanding 5. Specialized tissues can develop by cell differentiation in multicellular organisms. Differentiation is a process where cells become more specialized and distinct to other cells. Understanding 6. Differentiation involves the expression of some genes and not others in a cell's genome. All cells share the same identical genome in an organism. The activation of different gene sections give chemical signals and allows cells to differentiate, which means identical cells can become two distinct cells, one as a neuron or another as an epithelial cell. This is done by the activation of different gene sections, specifically the way genes are packaged. Active genes are packaged in a loose form as euchromatin, so that the transcription of genes can happen. On the other hand, inactive genes are packaged in a tight form as heterochromatin, so it saves space and not to be transcripted. Understanding 7. The capacity of stem cells to divide and differentiate along different pathways is necessary in embryonic development. It also makes stem cells suitable for therapeutic uses. Stem cells are important because of its two key properties. First is self-renewal. Stem cells can continuously divide. Second is potency. Stem cells have the capacity to differentiate and become specialized cells. Stem cells play a big role in embryonic development as all specialized cells root to the stem cells. But stem cells are also important when the non-stem cells are damaged and have to be replaced. Application 3. Use of stem cells to treat Stargardt's disease and one other named condition. Stem cells are used to replace damaged cells with healthy ones. The process starts with using a biochemical solution to trigger stem cells to specialize into desired cell type. Then the cells are implanted into the patient's tissue by surgery. At this moment, suppression of the host immune system is required to prevent rejection of cells. And lastly, it is important to monitor new cells so that they won't become cancerous. To give some examples, Stargardt's disease is caused by gene mutation, which triggers vision loss by damaged retina cells. It is treated by replacing the dead cells in the retina with functioning ones derived from the stem cells. Parkinson's disease has symptoms of tremors, slowness of movement due to the death of dopamine-secreting cells in the midbrain. Since dopamine is responsible for smooth movements, it is treated by replacing the dead cells with cells that produce dopamine. Leukemia is a type of cancer where abnormal large numbers of white blood cells are produced. White blood cells are produced in bone marrow. Hence, in order to cure leukemia, the cancer cells producing excessive white blood cells must be destroyed. Large needle removes fluid from bone marrow, which includes stem cells, and it is stored by freezing. Then the patient undergoes high chemotherapy drugs to kill all cancer cells in the bone marrow. Lastly, the stem cells are returned to the patient's body and re-establish themselves to produce both red and white blood cells. Application 4. Ethics of the therapeutic use of stem cells from specially created embryos, from the umbilical cord blood of a newborn baby, and from an adult's own tissues. Stem cells can be derived from embryos, umbilical cord blood of a newborn baby, or adult tissues such as bone marrow. Although there are 3 sources, the greatest yield of pluripotent stem cells come from the embryo. However, an ethical concern arises since it usually involves the death of embryos when stem cells are taken. But are embryos a human being? When will human life begin? Such questions are debated, whether killing the embryo and gaining a stem cell is ethical or not. Another argument is the creation of stem cells by in vitro fertilization, IVF. It is considered unethical to create human lives for the purpose of obtaining stem cells. Additionally, if women will be paid for supplying eggs for IVF, it can lead to exploitation of young females. But it is important that therapeutic use of stem cells have potential to treat diseases and disabilities that are currently incurable. Skill 1. Use of a light microscope to investigate the structure of cells and tissues. Since it is difficult to explain the Skill 1, I would like to recommend a video from Amoeba Sisters, Microscopes and How to Use a Light Microscope. This video will really help you to understand, so if you are interested, I posted the link below in the description box, so please click it. Skill 2. Drawing cell structures as seen with a light microscope. There are 3 rules when we draw cell structures. First, use a sharp pencil and draw it in a single sharp line. Artistic students tend to draw it in several lines like this, but it is better to draw it in a single line, so be careful. Second, join up lines to form continuous structures. It is important to make the cell's structure simple. Third, draw lines freehand, but use a ruler for labeling lines. If you keep these 3 rules, you will definitely get a good grade for drawings. Skill 3. Calculation of the magnification of drawings and the actual size of structures shown in drawings or micrographs. In order to calculate, it is important to remember the formula. Magnification equals the size of image divided by the actual size of specimen. For example, if the length of an image is 30 mm and represents a structure that has an actual size of 3 micron, where you have to find the magnification, you first must match the unit. 30 mm is 30,000 micron. Hence, by plugging it into the formula, it becomes magnification equals 30,000 micron divided by 3 micron. Hence, the magnification is 10,000 times. Calculation might be difficult, but try to start by remembering the formula. Today, we walked through the IB Topic 1.1 Introduction to Cells. I hope this video was useful and helpful for those of need in Bubbly Bio. Thanks for watching, and if you have any requests for topics of contents, please comment below. And lastly, I would be really happy if you subscribe Bubbly Bio. See you guys in the next video.